Thank you so much for coming out on this rainy morning. I first want to thank all of you who are supporters of the Cancer Research Institute. It's really because of you that CRI can carry out its mission and really support world-class immunological research all around the world. And so this morning I look forward, I hope, to an exciting discussion that will be informative to all of you. And as you know, the theme of the talk today is really about redefining cures. So I think we're going to hopefully learn that the new developments are really challenging the way that we traditionally think of cancer treatments, and it's enabling us to really view cure in a different way, and that's kind of what the take-home message today will be. So I first of all also want to thank our panelists here who are all CRI-supported scientists, and we're very lucky to have them here, and I thank you for taking the time to do this. So I'm going to make some brief introductions, and they have so many titles and things, I want to make sure I have it right, so I'm going to be reading this. First we have Dr. Bob Schreiber, who is an alumni-endowed professor of pathology and immunology. He's a professor of molecular microbiology and the program leader in tumor immunology at the Siteman Cancer Center, all at Washington University School of Medicine in St. Louis. Bob's a distinguished immunologist and a laboratory scientist who has turned into a tumor immunologist. And he really re-examined and subsequently really reshaped one of the basic tenets upon which the whole field of cancer immunology is built. So we're very happy to have you here, Bob. Then we have Dr. Nina Bardwaj, who is a professor of medicine, pathology, and dermatology, and the director of the tumor vaccine program at New York University School of Medicine. She's also another major figure in the field of human cancer immunology, and her work really focuses on the biology of an immune cell called the dendritic cell, and using them and other strategies to develop vaccines and other cancer immunotherapies that target cancer. So thank you, Nina. And then Dr. Jed Walchuk, who is an associate member and the director of immunotherapy clinical trials and an associate director of the Ludwig Center for Cancer Immunotherapy at Memorial Sloan Kettering Cancer Center. He's also an associate attending physician at Memorial Hospital and an oncologist who specializes in melanoma. Jed's a clinician scientist who has led some of the most exciting clinical trials of new cancer immunotherapies, and some of which have produced very remarkable patient responses and durable remissions, which we'll be hearing about in a little while. So now before we kind of jump in to some questions and answers here, I just thought I'd set the stage and make sure we're all kind of on the same page of understanding here, and that I think you all know that cancer is really a term that describes probably 150 to 200 different diseases. They're all different, but they all share some basic characteristics, such as uncontrolled growth and the ability to really invade outside their normal tissue parameters. Unfortunately, today in the U.S., 1.5 million Americans will be diagnosed with cancer, and 570,000 will die from it, with lung, colon, breast, pancreatic, and prostate cancers really the top cancers in the U.S. Now, there's been an explosion of our understanding of cancer in the past 40 years or so, but I'd say, unfortunately, in this same period, the way we treat cancer has really not changed to any great extent. There's still 3 primary ways that cancer are treated, and this is surgery, radiation, and chemotherapy. We all know, however, that these treatments have limited effectiveness, really depending on what extent the disease is at the time of diagnosis and what type of cancer it is. So there's obviously a new need for new approaches to how we really handle cancer. And immunotherapy is really what we're talking about today. And we all know that I think what we're learning about immunotherapy and how it's being used is helping us to kind of redefine what we call cure. I think we all want to cure cancer, but I hope that today you'll see, based on the science that we've learned and what we're seeing that the immune system can do, that cure may look different from what we have traditionally thought it to mean. And that, namely, controlling cancer and turning it into a chronic disease that we can live with may be the new cure. So that's kind of the take-home message and the theme for today. So we'll get started with some questions. And Bob, I'm going to start with you. So I feel like Oprah. First, a new understanding of cancer within the immunological context. You know, when the Cancer Research Institute was founded in 1953, there was the belief that the immune system could recognize and destroy cancer. And thanks to your work and your colleagues, that's no longer a hypothesis, but rather a truth that's really grounded firmly in good science. So can you kind of take us through what are the pivotal findings that have allowed us to really now understand that the immune system can recognize and control cancer?
Certainly. Well, the idea that the immune system can control cancer is actually a very old idea. It started probably over 100 years ago. The difficulty then was we knew very little about immunology, and so it was, one could not pursue it. In the late 1950s and early 1960s, work from a professor here, Lewis Thomas, and McFarlane Burnett in Australia, came up with this idea of giving it a name, and the name was called cancer immunosurveillance. Again, it was a really good idea that the immune system would be able to detect developing cancers and destroy them. We would call that a no-brainer today, but the interesting thing was at the time there really wasn't any experimental evidence to support the cancer immunosurveillance hypothesis. The problem with proving it was in the 1950s and 1960s is there weren't good models of immunodeficiency. And so one could only work with a single model, a mouse model that was a partial immunodeficiency. And when it was used to test whether the immune system could detect cancer and destroy it, there wasn't any experimental data that said that it did. So this idea of cancer immunosurveillance was actually discarded and lied dormant in most places in most laboratories for many years. A couple groups continued to work on it. Lloyd Old always believed that it existed and continued to push on that and to recruit people to look at these issues in a more detailed manner. I was one of his recruits. And by the time I started to work on it, our ability to control the immune system, to work with immunity, to have models for the immune system got much better. We had antibodies that we could deplete immunity. We had mice that had clear 100% immunodeficiencies. And so one could now go back in and really reevaluate this sort of discarded idea. And like many old ideas, when you apply now new technologies, you realize that there was a lot of truth to it. And so we started to perform these experiments in immunodeficient mice and showed that indeed they developed tumors more rapidly and in higher numbers than their wild-type counterparts. And this work was eventually repeated by a number of different laboratories. And I think in general now the idea that the immune system can control cancer has come back into vogue. With an acceptance of this, you can now move forward and you can look into the details of how this works. And that's been basically what our work has been focused on, taking this idea that the immune system can control cancer and looking at it in detail. And by doing that, what we found is that the immune system does two things. It controls cancer, so cancer immunosurveillance occurs, but a cancer that escapes the immune control has actually been shaped by its battle with the immune system and comes out a stronger cancer than it did going in. And so we realized that maybe some of the problem was the term cancer immunosurveillance, which implied pure protection. And so, like many things, we renamed it and called it cancer immunoediting to stress this dual host protective and cancer shaping or editing function of immunity. What's come out of this work has been very interesting. An understanding of how the immune system sees developing cancers, what kind of mechanisms it uses to destroy these cancers. Yet we also realize that sometimes cancer can escape the immune control and they enter into a second phase that we've called equilibrium. So the first phase we've called elimination. The second phase we've called equilibrium, and this is a time of tumor dormancy of which the immune system can't destroy the few remaining cancer cells, but they keep them in check. They stop them from growing out. And in many cases, this may be the endpoint of the immunoediting process. A cancer may be kept in control, and we may live with cancer for our entire lives. So cancer may become, as Jill said, a chronic disease. At some point, it might be that the cancer cell is shaped by its battle with the immune system and then exits the equilibrium phase and eventually escapes immune control. So that would be the third phase. And so we've cleverly named this the 3 E's of cancer immunoediting. As this was being developed, I'm almost done.
Okay. I'm going to cut you off soon, Bob.
Okay. As this was being developed, one of the Cancer Research Institute meetings, Jill got up to give the final closing message. And in her message, she brought up the idea of perhaps using the immune system to control cancer as a chronic disease, rather than, if we can't cure it, maybe we can control it. And it fits so beautifully into our 3E model, because this is exactly how we think of the second E of equilibrium. We think of it occurring naturally, but of course it can occur therapeutically. And the idea would be, if we can learn what the mechanisms are of natural equilibrium, as our colleagues in the clinic begin to get better and better in figuring out how to use the immune system therapeutically, then perhaps the immune system can actually be used to create the second E therapeutically.
So I think this leads right into kind of opening this up to everyone else on the panel. It's really, so how are we really integrating this concept of immune-mediated balance between the patient and the tumor? How is this, in the clinic, actually in patients, are we seeing that this has actual application? And so I throw this out to those that treat patients.
So I think that in the past few years, we've actually gained some very deep understanding into how true Bob's observations have been. We of course believe the science from the experiments that were done, and these were really groundbreaking results that the field was changed by in a very good way. But now that we have medications to treat cancer using the immune system, we have validated these observations in people. And I'll give you some examples of this. There's a drug that was now in final stages of development that was actually invented by Jim Allison, who's one of the leaders of the CRI effort, and in fact my boss also. And Jim developed a drug with the unpronounceable name ipilimumab, and it is in the last stages of FDA review for treatment of advanced melanoma. And what we've seen in treating now almost 6,000 people around the world with this drug is really the human examples of the 3 E's. There are some people, albeit a small number, maybe 10% to 15% of people who have true elimination with this drug, which turns on the immune system in a very global way to hopefully see cancer better, where tumors that were there before just go away. And that's clearly elimination. But then we see maybe another 20% of people or more who have equilibrium. People in my practice who got treated with this drug 4 years ago. Their scans were abnormal then, lumps in their lungs and their liver, but they're no different now. They feel fine, but their scans look like someone who's quite ill. But the balance between the patient and the tumor has been restored. And except for the inconvenience of being reminded that, there's something abnormal on their scan, they wouldn't even know that they were unwell. And then there's escape. There are people who don't benefit from drugs like this. And there, we once again return to the laboratory to try to study tumors that don't respond to this particular immunotherapy, to identify what pathways go wrong in those tumors and hopefully develop avenues of therapy for those patients.
So, Nina, can you give us some examples of other obviously we see this, I guess the viewpoint is this equilibrium. Many people that are not sick may be in equilibrium. I think that's, is that a well-held idea nowadays, that cancers are developing all the time in many people, and the immune system is eliminating them or keeping them at a level that they can't be seen in check or having any ill part of them in terms of this?
Certainly as an immunologist and based on the work that both Bob and Jed have done, I would agree with that. I would say that the immune system can certainly do a good job keeping tumors in check and even eliminating them. And most of the evidence does come from animal models, but clearly from the work that Jed's done, we can see clearer cases of tumors being totally eliminated when the immune system is manipulated. And so we've focused in our own group on trying to understand how to at least boost the immune system in circumstances where, for example, as Jed pointed out, this particular antibody may not work so well. And his studies have provided clues that the patients who do better are the ones that tend to have better immune responses to begin with. So if we could try to understand how to boost the immune system even further, perhaps we'd be able to give these patients an opportunity to allow their immune system to work in conjunction with these other interventions to either eliminate their tumors or reach a point of equilibrium. And so we've worked a lot with various vaccine platforms that are designed to stimulate the immune system. And in our own case, we've worked with dendritic cells that Jill talked about earlier, which are very potent white cells that are key for arming the immune system initially with antigens like cancer antigens. And these dendritic cells, which are present in every single tissue of our bodies, acquire these antigens, become activated, and then go around basically arming the rest of the immune system. And so one thing we're trying to do is to learn how to take advantage of these properties of the dendritic cell to boost the immune system. And so our vaccines are designed to either isolate these dendritic cells, give them cancer antigens and give them back to people, or use indirect methods to activate them within the body itself.
So we're in terms of different therapies that are being developed today that are based on the immune system. Can you kind of talk about what are the key pieces of this, or what are the areas most exciting that we have to really focus on, you think, that have the most promise?
Well, so I think we've hit on two of them so far, two specific examples of vaccines that in a very focused way get the immune system to recognize a particular target. We know that vaccines alone can do that. They can generate an immune response to targets on cancer, and it's really been primarily through the efforts of the Cancer Vaccine Collaborative, which is a joint program of the CRI, that we have gained enormous insights into how to induce immune responses to specific targets on cancers in people. But we know that those immune responses need to be more potent than a vaccine alone can initiate. And this is where these other more global immune modulating drugs like ipilimumab come into play, and forming combinations of both drugs that sort of turn up the volume on the immune system in a general way, but also focus it on a particular target on the tumor is really where we want to be. Ipilimumab is a first-in-class kind of drug that does this, but there are now other drugs that are entering the later stages of clinical development. They don't really have names yet, let alone unpronounceable names, but they attack pathways such as the PD1 pathway, which is if you imagine that a tumor cell sees an attacking immune cell and it's trying to figure out a way to defend itself, the most clever way that a tumor cell can do is to cause death to that attacking white blood cell. And so this is what the PD1 pathway does. The tumor cell puts a molecule on its surface, which if that molecule contacts an immune cell, the immune cell dies on impact. And so now we have a drug that blocks that interaction. And that is showing some very early promising results in melanoma, in kidney cancer, and in lung cancer as well. And so we are really now seeing that the science, such as the studies done by Bob and Dr. Old, is absolutely directing the medicine. We now can capitalize on the insights that were made based upon decades of basic science to develop therapeutic interventions that aren't generated by happenstance. This is not an accident that we have ipilimumab and that we have anti-PD1 drugs. These were drugs that were specifically developed based upon the molecular mechanisms of the immune system that we've spent so long to learn about.
So that kind of brings me back to the thought of this whole continuum of supporting research. Well, why support basic laboratory research for so many years? How do you get it into the clinic? We're now seeing actual translation of your discoveries and seeing that they're proving out not only in mice, but in humans. And what about the crosstalk back? I mean, is there a lot of interaction now that you have actual patient samples, we're seeing what's happening in patients? Is that coming back into the laboratory? Is there new research being done now based on what we're learning in the clinic so that there's this two-way street going on?
So I maybe can say a few things about that. One of the things that's coming back is the understanding, a better understanding of what the targets are in terms of what the immune response is seeing in the tumors. From cancer biology, we're now learning that tumors actually have a number of abnormal proteins. They're either expressed abnormally or they have mistakes in them. And these actually, many of them can function as the targets for an immune attack. We don't really know exactly which are the best ones. What's interesting is when one looks at these patients who are being treated with things like ipilimumab, you can not only see, as Jed mentioned, that the ones who do better are the ones that have pre-existing immunity, but also that there's often an uncovering of new specificities. And those specificities added on to the ones that are already in existence help the immune response be more effective in terms of these tumors. And so we're learning at the laboratory bench by looking at what's going on in the serum and in the blood of the patients, basically what the immune response is seeing and which of those targets are the best ones then to focus on in the future.
How about you, Nina?
Yes, absolutely. We're taking, in our case, patients who've completed trials that have been supported by the CVC with the platforms that I mentioned, and like Bob, taking their serum and their cells and trying to understand what they respond to following vaccination. And that's giving us a lot of information. We're learning, for example, what targets the T cells, for example. These are effector cells in the body that are responsible for eliminating or controlling the tumor. We're getting a very good handle on what they see and how they function. We're also getting a better understanding of, once these cells do begin to respond and kill off the tumor, the concept that with the tumor cell death, a release of new targets that can be picked up by the immune system, and so a new wave of immune cells can be generated that can effectively contain the tumor, something we call epitope spreading. And we're learning from studies that, again, we do with the CVC by examining the sera, the breadth of an antibody response, and beginning to understand how important the antibody might be in containing the tumor as well.
This is definitely a two-way street. The idea of the bench to bedside direction certainly is true, but we are most frequently actually taking things back the other way. Specimens, tumor, blood, etc., from patients and studying them in the laboratory. And this sounds like an awfully simple thing to do, but in the day and age that we live in of scrutiny of what happens to human biospecimens, this is very complicated. Spend an awful lot of time schlepping samples across the street myself, and waiting outside operating rooms for very valuable tumor samples so we can study them in the laboratory. And and we don't know when these things are going to happen, and we have to, chance favors the prepared mind, but also the prepared hands who happen to be waiting outside the operating room. But we gain so much from studying single patients. I mean, this is something that Dr. Old taught me for a long time, that we cannot pass up the opportunity to learn as much as we can from single people, because that is really where most of the most important observations that we've made have come from. So we are very much full-time engaged in this bi-directional interaction between the laboratory and the clinical setting.
So what what do you really think the future of cancer treatment? Where does immunotherapy fit in your view, in the next 5 years, 10 years, with surgery, chemotherapy, and radiation? I mean, is it going to become a standard of care that every oncologist is going to look to as an option? Is this still going to be experimental and only happening at teaching hospitals? How do you see this falling through and coming out?
I think 2010, 2011 is really when cancer immunology becomes, the fourth standard way to treat cancer. I think that there have been two, milestone events in the past year. One is the approval of SipuleucelT or Provenge for
These names,
prostate cancer.
SipuleucelT.
We're trained not to use the brand names, so we have to use the less less eloquent chemical names. And also the positive results with ipilimumab. I think that those are very important validations that this is now conventional therapy.
I would agree. I think SipuleucelT, otherwise known as Provenge, which is easier to say, is the first cell-based approved, FDA-approved immunotherapy in this country. And that's a real milestone. And that particular therapy takes advantage of dendritic cells, which we mentioned earlier, these key immune activators in the body. And while there is some improvement in survival in patients with prostate cancer who get this therapy, the fact that it has been approved, I think, as Jed mentioned, is a real milestone for the field. And this is by no means a home run, but what it has done is actually open the door for those of us who work in this field to create even better therapies. So I think we're going to see amazing improvements upon that platform. And what we're also going to see is combinations of therapies. So for example, a vaccine together with IPI, I can't say the name, so I'll use the short version, and very exciting new combinations in the field that I think are going to really make major inroads into the treatment of cancer. And so I'm a firm believer that immunotherapy is going to be part of the general approach that oncologists will use to treat patients who are otherwise, resistant to other forms of conventional therapy.
I would also add, I think the one beautiful thing about immunotherapy is it's not an either-or situation. That is, you don't have to make the decision if you treat with immunotherapy versus surgery versus radiation therapy. In fact, in many cases, I think what's showing up is that it works better when they're together. Mm-hmm. So if you remove the bulky tumor by surgery or you irradiate, that process alone releases some of the dead tumor cells that actually can go on to activate the immune system and give you a better chance of immunotherapy, especially with something like ipilimumab. So perhaps as the cleanup hitter, the immune system is the perfect tool.
So I think, from what you're saying, I guess because also immunology can lead to memory. We all know that childhood vaccination, we get vaccinated, you don't get that childhood disease if you're exposed to it because the immune system has memory. So how does memory, you think, play into turning cancer into a chronic disease that can be controlled as the ultimate cure?
Well, maybe I can start that discussion. What we know from the model of equilibrium in the mouse is that, in fact, it is the adaptive portion of the immune system that is driving this controlling force that keeps these tumors from expanding, or these tumor cells from expanding. And that essentially is what memory is. It's the adaptive immune system, the T cells and the antibodies that are made. And so I would argue that, in fact, this is what's probably also happening in terms of immunotherapy as well. You get good memory. If you have some residual cancer cells, your immune system, your adaptive immune system, because of its incredible specificity and power to control these tumor cells, that keeps them in check.
And I think that's where vaccines become important, understanding how to generate that kind of memory so that if a tumor should recur or if there is residual tumor, that kind of immune response can be manipulated or generated to keep tumors in check. And I think that Bob's data and his experiments have allowed us to now appreciate that we can achieve goals of equilibrium and keeping cancer in a chronic state if we're at least not able to totally eliminate it.
I think that this memory gives immunotherapy and responses to immunotherapy a durability that is not seen with other forms of cancer therapy. And again, I'll return to the field I know best, which is the treatment of melanoma. Just to give you a few examples. The patients who respond to ipilimumab and also another immune therapy called IL-2 or interleukin-2, which was actually approved for melanoma about 12 years ago. If those patients are in remission for 2 years with IL-2, they're still in remission 10 years later. And we don't see anybody relapsing. And ipilimumab is a newer drug, so we maybe only have 5 years of follow-up. But the vast, vast majority of people who have some degree of control, whether it's equilibrium or elimination from ipilimumab, don't come out of remission. This durability is a hallmark of immunotherapy, really because of what Bob and Nina just said, that we know that when we're young, we're vaccinated, and our immune system doesn't forget those vaccines very easily. And so this is really what you want in protecting against recurrence of cancer as well. And if we put that in comparison to another very new, exciting way to treat melanoma with a family of drugs that block a gene called BRAF, which is mutated or changed in 50% of melanomas. These have turned out to be extremely powerful drugs to cause remissions in the vast number of people who are treated with them. But these remissions last on average 6 or 7 months. And so really the way that we see immunotherapy moving forward in its most impactful way is in combination with other forms of cancer therapy to deliver that durability. Because that is really what most people are looking for. They don't just want to stop the cancer from growing for a short time. They want to live a normal life for as long as possible. And I think this idea of combinations of immunotherapy that provides that durability with other types of cancer therapy is where the field is going.
And so I think, I don't know where to end. I mean, hopefully we've gotten across a lot of this. I think we're going to open it up to the floor for some questions soon. But maybe I'll just start off in terms of where, being an organization, Cancer Research Institute, that supports science. Where, this may be a tough question to throw out, but if I had $25 million to give each of you, where would you think that's best spent in terms of delivering on getting treatments that are ultimately going to benefit patients? No, it's a tough question, but I won't start with you. You give me this look. I bet that start with you. Nina?
Okay. Well, I would use part of the money anyway to really improve our vaccine platforms. I think we have a lot of exciting new platforms that have come into the clinic now, and one I mentioned is Provenge-related to dendritic cells, but the others are natural adjuvants that we've derived from pathogens that we can use to stimulate the immune system. And combinations of those adjuvants, for example, I think will be key in understanding how to generate the kind of T-cell memory that both Jed and Bob talked about. And then I would really like to see some of these funds used to test some of the ideas that Jed talked about. How can we combine vaccines with these other agents that are designed to improve T-cell memory? And so to see some really good studies that combine those two approaches. And in addition, expand them to include very exciting new drugs. Jed talked about BRAF inhibitors, but also modalities that are currently also in use that we now understand stimulate the immune system, such as radiation therapy, such as chemotherapy, which we thought for a long time were rather sort of innocuous therapies, but we now know can in and of themselves stimulate the immune system. So I think the future will be trying to pull all these modalities together in a very organized, systematic way, taking advantage of what we know about the immune system, what we've learned from all the studies that have been done in both Jed and Bob's labs, and making very targeted approaches to treat cancers. And they might be different for different kinds of cancers or the stage of cancer, but I think we have a whole new understanding and a wealth of just agents and opportunities now that I believe in the next decade we'll really be able to move forward very, very fast.
I just throw my vote in again for exploration both in the laboratory and especially in clinical trials of combination therapies. The way to sequence new drugs together, what do you give with what, in what order, and really doing in-depth analysis of what's going on in the patient when you're combining new drugs is where we need to be. If we think about medicine's greatest victories in the past century, it's really the toughest nuts to crack have been HIV and tuberculosis. And I think both of those endeavors have taught us the important lesson that combination therapy is what ultimately yielded the most benefit. And I think here we are seeing, really through Bob's work, the need to expand upon elimination and equilibrium and really look at why we're getting escape. And I think that the idea of treating cancer by treating the tumor needs to be expanded to a vision where we treat the patient and the tumor at the same time. And for the first time, for diseases like melanoma and other cancers where we are learning about the targets that are important, we can actually, with precision, treat the patient and the tumor. And I think that is where we're going to realize the most benefit.
Just to follow up on that, I think an interesting thing that people might want to know about is the treatments in melanoma that we're seeing with immunotherapies, that unlike chemotherapies that you see responses immediately. Like when you get a chemotherapy, most patients are looked at after 12 weeks of treatment. And if the tumor hasn't shrunk in 12 weeks, I think the oncologist says, well, this treatment fails and we have to move on to something else. And I think we're now seeing that with the immunological treatments, this is not the standard that has to be looked at anymore, because the immunological treatments are not directly affecting the tumor, but rather are treating the patient. And the patient's immune system needs to get revved up to then react to the tumor. So I think, just if you want to just talk about that a little bit.
I think our expectations as human beings to see something happen on a certain time schedule and quickly. Of course, when someone is battling cancer, they want to see results quickly. We need to rewrite the rules by which we judge immunotherapy as an anti-cancer therapy, because as Jill alluded to, with immunotherapy, we're treating the patient and the patient is treating the tumor. And that may happen quickly in some people. It may happen slowly in other people. In fact, people treated with immunotherapy can, in fact, look worse before they get better. And so we, as a community of immunotherapy investigators, have really developed new criteria for judging the results of these novel therapies that incorporate the biology, really building on the science to inform clinicians as to when not to abandon therapy. Because it may still work. And in fact, ipilimumab may have actually ceased development a few years ago were it not for an appreciation of the biology between patient and tumor that basically caused the drug company and the FDA to say, wait a minute, let's change the endpoint of this trial to not look at whose tumor shrinks at 12 weeks, but rather how long is the patient living? Because that's really the most important thing.
So anything else you want to add, Bob?
Yeah, I would add two other things. One, in terms of the ability of the immune system to either control or destroy cancer. The immune system is a very complex system with many, many different mechanisms to destroy foreign cells. And we know that many of these components are play a critical role in any of the 3 E's that we've talked about today. But what we really don't yet understand is the precise choreography in which they play their roles. And I think at the basic level, we need to learn more about this and what cells are coming in at what time and what molecules are being involved at what time. This would probably help us understand why tumors sometimes get larger before they shrink because of the infiltration of these different types of immune cells. And I think learning more about that will help us in the future also improve our immunotherapies. I think the ultimate goal of immunotherapy, talking as someone who's never treated any organism with 2 legs, is personalized. I would say personalized immunotherapy would be the best, to really focus in on the immune system of the individual, as Jed mentioned, the unique properties of the tumor, perhaps, not only the antigens that it might have in common with other tumors like it in other people, but the unique antigens that might also be present in the tumor. Maybe we can make immunotherapy even safer than it is by looking at something like that in the future.
So I think now I'll open up to the floor for questions. Peter?
So I have 2 questions that are related. One, just exactly what you said, which I was going to ask about, is everyone's been talking about personalized medicine. How far are we from being able to characterize the individual nature of it?
This idea of personalized immunotherapy for tumors, we've gained so much information from the Human Genome Project, and part of the benefits of all of that effort has been the generation of a huge machine across the country, across the world, that we can now sequence the entire genome of anything we want in a very rapid period of time. 2 years ago, the first full genome of AML, a leukemia, was published. It was done by my colleagues at Washington University. It cost $2 million to generate that sequence, and a long time. Now, because of the advances in the field, one could do the full genome of a tumor for tens of thousands of dollars. And more importantly, one could do the sequence of just the expressed genes in a tumor for $2,500 in 1 week. That's a remarkable accomplishment. And what it allows us to do as immunologists is to look at the sequence of every gene that's expressed in that tumor cell, compare it to the normal sequence of genes, and then identify the mutations, and perhaps from there take it to the next step and figure out which of those mutations might be the targets of the immune system, and then rely on people like Nina and Jed to target and make vaccines against those antigens, and then together with perhaps ipilimumab, generate a personalized vaccine. So I think we're now entering a phase where this is suddenly possible, where just 12 months ago this was not. And we're very excited about this. We're doing this actually now in our mouse models to see if this can be really achieved. Some of my colleagues at Washington University and elsewhere are also doing this with human tumors as well.
What was the second question, Peter?
The prophylactic versus
Oh, you might want to talk.
Well, I think, in the future we probably will see prophylactic vaccines. We have a lot of work to do. And there are, besides Cervarix and Gardasil, there are, the HBV vaccine, you could think of as also a prophylactic vaccine. That it would result through prevention of Hep B infection, consequently downstream liver cancers. So, and then there are other examples where there are now vaccines that can eliminate preneoplastic sorts of conditions. So one of our colleagues, for example, has successfully treated a condition called vaginal epithelial neoplasia with a vaccine. This is a very resistant pre-malignant condition. So we're beginning to see certainly many examples now where prophylactic vaccines can be effective. I think the issue is going to be, in terms of targeting different cancers, we're going to have to understand who's at risk, what the antigens are, and before we implement something very broadly. So as we move forward with personalized medicine and we understand the genetics of individual tumors and individual people in terms of mutations that might put people at risk for certain tumors, we're going to get better at designing those kinds of vaccines. So I do think that's going to happen. It will need a lot of work, obviously, and a lot of research.
Thank you.
Can I add one other thing? Sure. One of the other interesting things that's coming up from tumor biology is the subset of cancers that are virally induced by oncogenic viruses. At one point, this was considered to be a very, very small number. It seems to be growing, and as we go along, we hear more and more reports of strange viruses that seem to be associated with certain kinds of cancers. We don't know how extensive this will eventually become, but certainly for those cancers which may be actually have a viral basis, then prophylactic vaccines becomes entirely possible, because this is just vaccination against an infectious organism that we've become quite good at. And so for those subset of cancers, I think that that's a very good possibility.
And I should add that the disease I mentioned, the vaginal intraepithelial neoplasia, is actually an HPV-associated premalignant condition. And so that's just to support your argument, a virally associated cancer that was very effectively eliminated.
Yes.
I wonder if you comment on this and how you could reconcile it. I've had some personal experience, as you did, of actually taking a tumor. I'm not a doctor. Taking a tumor from the surgery room to down the block, across the street, actually across the ocean, to have it treated in a dendritic cell experimental program.
I think that sea change is actually already beginning to happen. And I think any of the newer therapies that attack mutated pathways in a cancer cell have now absolutely demanded that the tumor's genome, at least parts of it, be sequenced in real time to determine therapy. And I think that we have a few examples of this. RAS mutations and RAF mutations in melanoma. The drugs that block RAF in melanoma do nothing. In fact, they may even potentiate the growth of the tumor if it doesn't have the mutation. So it's now become absolutely imperative to do reflexive genetic testing of the tumor once the patient's diagnosed. But this is a big change, because this is a philosophical shift for pathologists, where all of a sudden, they look at a glass slide and that tells the whole story, to now, I'm sorry, you have to give up part of that tumor and send it to a different laboratory and let someone extract the genes from it. And the extraction of those genes and the elucidation of what's gone wrong in that tumor will allow for the construction of a personalized approach. We may not need to send that tumor to be made into a vaccine anymore. We just may need the in silico data, if you will, of what's wrong, and then a customized vaccine can be constructed. So the world is changing right now. We're still feeling the stresses and strains of it. I mean, it takes us 3 or 4 weeks to get knowledge about the mutation of one single gene inside a tumor, when in fact the work could take a few hours. A lot of that is just getting our infrastructure ramped up to deliver information on a real-time basis, but it is happening.
And I would add that SipuleucelT, which is the first cell-based immunotherapy approved in this country, is very individualized. It requires that each patient have their dendritic cells and other cells removed through a process called leukapheresis, and then the antigens added, and the cells are then reinfused back into the patient. And so there's an example of very individualized therapy that has come to full approval. So I would agree with Jed, it's happening.
Other question?
How do we compare in terms of regulation and ease of movement in places like Holland or Israel or Australia? I mean, how much more difficult is it for you to be able to, or how much easier is it for them?
I think it's a great question. I think for an exercise like getting genetic information out of the tumor on a real-time basis, it's not that much different here. It's more just the changing world of pathology, of folks who were not used to working under time pressure of, what you deliver to the pathology laboratory is actually necessary to determine the therapy, to a very high degree of precision. I think that it's an easier problem to solve. It's actually just building in new layers of people and processes in pathology departments, and building new laboratories to really do this type of high throughput genetic analysis, which is really happening. I mean, in the past 2 or 3 years, it's better. It's not great yet, but it's certainly better.
Where is it better? In here, in Boston?
I think it's better all over. And I think what's also happening is that drug companies are realizing that this is the future. And so they are now building in what are called co-diagnostic tests with their drugs to say that not only are we going to make the drug, but we're going to make the test to determine whether the drug is for you or not. And that's very important because that is going to speed up the process as well.
Any other questions?
I have one simple question. Is there any downside to, if let's say you've already done chemo?
I think you can look at it perhaps in 2 ways. If one is treating without knowing what the antigens necessarily are, then you're walking a fine line when you're trying to enhance the immune system's ability to recognize and control a cancer cell, and the immune system's ability to interact with our own cells and cause autoimmunity. And we've come a long way, I think, in learning how to be pretty good at that. Ipilimumab, when it was first used, caused enormous kinds of autoimmune reactions. But now it's being used in a much more limited manner in terms of the dosing, and those reactions are much weaker. But still, there is this fine line that one has to walk between that. One argument for personalized immunotherapy, though, would be that if you're targeting antigens which are expressed only in the tumor and nowhere else, that perhaps the ultimate goal of immunotherapy would be to do that so that the autoimmune reactions might be considerably less. So I think that would be the ultimate goal. I think right now the fact is that one can control the autoimmune reactions by using very carefully dose-limited amounts of these agents.
And I would add that the CVC's done a lot of work in doing just that, taking antigens that have been identified within the tumor but not expressed in most normal tissues, and then developing a number of vaccine platforms that take advantage of these antigens and then immunize patients in what we call the adjuvant setting. So the tumor's been resected, they're disease-free, and then the idea is to try to boost the immune system to this antigen, which has been expressed in their tumors, in the hope that vaccination and generating good memory will control the tumor should it recur. And so there are many programs like that the CVC and the CRI in particular has funded, including a number of studies that we've done at our institution that are doing just what you're asking.
Question? Any other? No? Oh yeah, we're just on time. Okay, no more?
Maybe one thing that has not been said here that maybe should be raised is how important the CRI has been in all of this.
Mm-hmm.
I think we're all sitting here, I mean, because of the CRI. We've all been attracted by different aspects of them, of the programs that the CRI has built. They've really helped to elevate immunotherapy, tumor immunology into a very hard, critical science that is now moving forward. They support our fellows. They help train people. They bring us together to discuss between basic and clinical people what kinds of things we can best do together. They support the CVC and really make immunotherapy now with very hard and solid scientific readouts. And I think that there's very few organizations that one could point to that have done such a good job. And I would even compare the CRI to the NCI, the National Cancer Institute, which has for years almost ignored immunology. And it's been the pioneering efforts, I think, of the CRI that's really kept the idea of tumor immunology and extending it into tumor immunotherapy alive.
I completely
Yes,
agree. And
yeah.
I just to expand on that for a moment about the CVC, which is the clinical trials program, that this is an organization that is worldwide and has accomplished so much through the efforts of Nina and our other colleagues. And in fact, in the past year, the NCI has come to the same conclusion that the CRI did 10 years ago, that there is a need for a national network of immunotherapy clinical trial centers, and actually came to us and actually asked us to help them with this national network based upon the fact that we've been doing this on a global scale for the last 10 years. And so we received very positive feedback from sometimes very objective reviewers in the government about our efforts. And I think it again is validation of both the science and also the vision of this organization that we're so grateful for.
I'll just second that. I don't think I have much more to add other than to say certainly I couldn't have done a lot of the studies that over the past 5 or 6 years without support from the CRI. And one other little known fact is that the CRI also does a lot of education and outreach. They support, for example, a program, a winter immunology program that is held in India every couple of years to spread the word out to third world countries. So we're very appreciative of that support also.
Well, I think that ends us on basically everything that the Cancer Research Institute does could not happen without you and your support. So it all comes full circle. We thank you for supporting us for so many years. We hope you will continue, and we thank you for coming here. And I hope you've learned a lot, and I hope that we can come to understand that cure may actually be the new way. Control may be the new cure for cancer, and that we think that can be accomplished with immunotherapies. So thank you very much for coming, and we'll hopefully see you again next time when we do this again. So thanks.